I'm Jessica. I'm from Molecular Biology and Bioinformatics Lab from Professor Wilson. I'm here representing him to talk about genomics in sickle cell disease. It's a project which is being made with a collaboration of Professor Jack and Mark Romana and Professor Dimas. So here we are talking about specifically by sickle cell anemia, exactly on a phenotype called Hispanic sequestration, which Ana Cristina really explained and show what is. So briefly, the sickle cell anemia is characterized by a mutation on beta-globin gene, which causes a mutation, a change for a valine amino acid. And this turns a mutant protein, which will form long inflexible chains that is the opposite of the normal hemoglobin. And these chains will be stiff, you let the red blood cells stiff and angular, causing become stuck in small capillaries. So one of the most aggressive phenotype of this disease is splenic sequestration, which is the vaso-occlusion of the splenic venules, resulting in trapping of blood cells in an engorged spleen. So this causes, in most of the times, worsening of anemia and splenomegalia. What we have is enlarged spleen caused about the maintenance of the red blood cells, the sickle cells, on the spleen. So to understand that, we performed a whole exome sequence at the genome analyzer platform in 10 siblings pairs, being one of them with a splenic sequestration phenotype, and another one with the splenic sequestration phenotype. So we made a library with 55 base pairs, and after the sequence, the FASTQ values was first mapped with the BWA program with the reference, the human reference genome HD19, and then this mapped reads was marked and sorted to finally identify the variants with the GTK protocol, which involved the indirect realignment, the basic recalibration, and after that, the genotype likelihoods to test if the true positive variants and the false positive variants. After all of that, we made the functional notation for these variants, and we make a refinement to identify the possible variants causing this phenotype, the splenic sequestration. So the first result was about the mapping. Almost of the reads was mapped with 99% of mapping. And here we have the pairs, so the symbol pairs. So this one do not have splenic sequestration, and this one have the splenic sequestration. So far, so well. And all these reads was with a map quality more than 30, the 35. So here is the total coverage of the genome, the exome, sorry. So here at the epsilon axis, we have you can see the 90% of the exome was covered. And here on the x-axis, we can see that more than 50% of this exome was covered with 20 times of depth. So that was enough to detect the variants and to ensure high quality of that. So the total variants identified was 155,000 variants, about, and of them, about 30,000 variants was non-synonymous. And we made features by pathogenic prediction, like some programs as polyphane, safety, which will analyze the third D protein structure, and this with those filters, we generate about 5,000 variants, but only 882 variants was present in only affected samples. But none of the variants was present in all affected samples with this first filter. And here we can see we choose the 99.9% of trenches with a good cumulative positives, and with enough translation ratio, which is a method to identify the quality of the variant call. So, as we can see, the mutations, the number of the mutations on with transitions is much higher than transversions. And as you can see, especially the cytosine domain changes because the exome is really methylated. So this increases these potential changes at the exome. So, but this was as we were expected, and is a good measure to see if the mutations identifiers is good, right? So to identify the variants causative for the phenotype, we performed actually 3 types of analysis. But thinking about the gene, which we have the exons, we can have a common single variant which can cause the phenotype in all the patients. And we can have one gene hypermutated in a lot of exons in a lot of positions. And the other type is we can have a lot of genes mutations in the same pathway. So this can cause the disease. And we perform these 3 types of analysis to identify what can be the causative mutations for this phenotype. And the first filter was how we have the siblings. So the genotypes for them need to have different, right? So the first filter, we get only 1,641 mutations. And about that, we made the 3 types of filters which I mentioned before. So the most mutated genes, what about 15, get us the 120 mutations. So from them, we filtered by non-synonymous variants, which was only 14. And then for pathogenicity prediction, which was only 4, but none of them was present in all affected controls, in all affected samples or all in control samples. So because of that, we made another filter. And starting by non-synonymous variants from this number, so we get 177 mutations. And then we made the pathogenicity filter with 41 mutations, but again, none of the mutations were present in only affected samples or in only the control samples. And then we made the Heritance analysis, not based on pathogenicity, but with all the mutations. So we made the dominant analysis the recessive analysis and the condominant analysis for both, or the control group or the affected group, the splenic sequestration group. So we found two genes, the SOM gene, which a protein bind is a RNA, a genomic binding, and this gene is present in only the affected groups. And another gene is the RBMXL1, which is present at the control group. So when we look at the most mutated genes and analyze the pathways which they enriched, we can see the red ones, which is marked here, which is involved in mainly in cellular movement in reproductive system development and function. So for these genes, we can see they may involve the disease, but it is not a conclusive result. So a general result is the number of mutations per patient, which we can see at these bars, these blue bars. And at this heat map, we can see the most mutated genes, the 15 one, which are here deposited in the siblings pairs. So we can see there are differences between them, but there's not a pattern to say if there is exactly these mutated genes are really present in the same group of samples, right? Like control or splenic sequestration group. So further this, we analyzed the Heritance group, the Heritance analysis with the splenic sequestration group. So those marked red genes are mutated on the splenic sequestration group. And those genes enrich pathways like IL-10 signaling, toll-like receptor signaling, PPR signaling, involved mainly on immune response. So molecular networks, like we can see here, are enriched mainly on cell-to-cell signaling, and involved is involved on adhesion and this type of environment interaction, as Ana Cristina said before. And then when we enrich these genes with the control group mutated genes, we can enrich the histamine biosynthesis, which is involved in vasodilatation. So maybe really involved at the disease too. These pinky circles are the genes present at the mutated present at the splenic sequestration group. So here we can see that they enrich the cell-to-cell signaling interaction, as I said before, as the genes present only on splenic sequestration groups enriched too, and the hematological system development and function and immune cell traffic. So this was interesting result, but it's needed to study these genes, understand the function of them at the disease, and try to understand what is going on here, right? It's not a simple causative mutation, right? Is maybe a pathway or a lot of mutations involved at this disease. So we selected some mutations like at the gene MPAK-K4 and IL-1A, which they are involved mainly on oxidative stress, angiogenesis, inflammation, and metalloproteases, which are enriched at the environment of splenic sequestration and the bad function of the spleen. So after that, the next step is validate the selected mutations by single sequencing at these 10 patients which we sequenced, And then increase the number of the sequenced exomes, because it's only 10 samples, is a really small number to make a supervised analysis. And after that, we need to denotype the identified variations in a larger in a larger number of samples. And after that, understand what these mutations, how they are involved, and if they are associated with the disease, how they are involved with them. So thank you so much, especially here at Hemocentro as Professor Dimas, Simone, and Ana Cristina and Mariana, which was really involved in the project and helped us to understand mainly the biology of the disease, and the Professor Jacques and Marc Romanat from France. So, and also the funding agencies too. Thank you so much.